Welcome everybody to the next topic in the series for Da Vinci education. This is Dr. Andrew Heyman, and I am pleased to be part of this next topic, which I think is incredibly important for all integrative practitioners. This is part one of a two-part series in dealing with chronic inflammation due to a biotoxin exposure. For many of you, this might be a new approach and a new language in terms of assessment and therapeutics, but you will be surprised to see how many of your patients actually suffer from this condition. The first part of this series, I'll be discussing the evaluation process, which is quite extensive in this patient population. And in the second part of this series, we'll run through our treatment algorithms and how to deal therapeutically with these patients. So what is this chronic inflammatory response syndrome? You may have heard this before. I've had the pleasure of working very closely with the researcher and clinician who has done a lot of the science and development of this topic, which is Dr. Richie Shoemaker. Over the last several years, he and I have collected a lot of data on the subject and published a series of studies. So what I'm presenting today comes directly out of the work that we've done. And I'm passionate about this subject because it affects so many people. I'm passionate about it because integrative providers are very well suited for treating this patient population. And I'm passionate about it as well because it is very under-recognized as an entity, despite how common it is in the general population. So I think it's really important to put this knowledge and skill set into your clinical toolkit so that you can better identify these patients and also manage them effectively in your own practices. So this idea of chronic inflammation that's established has a certain set of criteria that we use to make the diagnosis. We know that the inflammation has to be present for at least 6 months. That's pretty typical and as criteria for any sort of chronic condition. It is inflammatory in nature, but it is a unique sort of inflammation, one that has sort of escaped notice of a conventional evaluation process for many years. And it's because much of the way that we measure evaluation in our patients focuses on a general inflammatory response. So we order analytes such as CRP or sed rate, or we focus on autoimmune conditions, and therefore we can run a battery of tests in that regard. It turns out that the inflammation that's generated in this particular population is located in the innate immune system, not the adaptive, and therefore typically all of our inflammatory tests are normal in this patient population. So we've had to develop a new language and a new approach to identify when an individual has this sort of inflammatory response. The defect that occurs, the reason why individuals have such a problem with this sort of inflammation is because of an inherited polymorphism on chromosome 6 that has to do with alleles that control the efficiency of antigen presentation. And it turns out that there are about 6 to 9 of these alleles that impair the efficiency of the handoff between the innate immune response, which is the early primal reaction to external threats through neutrophils and macrophages, as well as it includes upregulation of antigen presenting cells that will snip an epitope from the antigen and then in turn present it to a T cell. Well, what we found is that with these polymorphisms on chromosome 6, the antigen presenting cells can be pretty inefficient in that handoff. And so these patients remain stuck in this sort of early innate immune response where there's a storm of inflammation that's generated and they can't turn it off. The body does not have a mechanism by which it resolves itself back to normal. And so with this defective antigen presentation, this patient population is essentially consigned to chronic inflammation where the innate immune system remains upregulated and is not able to transfer the immune reaction to the adaptive side, which includes T cells and B cells with antibody production, and has a greater capacity for resolution of inflammation. As a result of this inflammatory process, it affects just about every organ and system in the body. And therefore, there are many, many, many symptoms that are part and parcel to this condition, which makes it incredibly confusing. So these patients tend to be very sick overall. Their typical presenting complaint is fatigue, but actually not always. And the inflammation and the secondary and tertiary physiologic changes reaches into just about every body system. So as a result of this, we had to build a scientific framework to better understand what we were looking at and how do we intervene in a way where we correct all of these issues. And there are many that have to be dealt with. So you'll see in part 2, there's a series of steps that you need to go through to get these patients better. And the foundations or the pillars upon which we build this science have to do with a variety of assessments and measurements that allow us to make the proper diagnosis. And this includes proteomics. So these are the very special inflammatory proteins and neuropeptides that you can measure in blood labs, as well as abnormal transcriptomics. So this has to do with the abnormal gene response that occurs primarily, but not only, in the mitochondrial gene set. And it turns out that these gene-related changes and loss of regulation is the core feature of why the inflammation is perpetual. And that is to say, the individual is vulnerable because of polymorphisms on chromosome 6. But once that inflammation is established, it itself influences hundreds of genes in the mitochondria, and that these gene pathways then turn on, and they tell the body to make inflammation, but they won't turn off on their own. That there's loss of regulation of gene response. These gene activity sequences will not go back to normal on their own. And so it really requires a therapeutic intervention to do that. As a secondary result, there's loss of regulation of the immune system. And as a tertiary result, we begin to see other abnormalities occur in various body systems. These can be hormonal imbalances, GI-related complaints, cardiopulmonary issues, chronic pain, problems within the nervous system, skin disorders. There is a whole host of changes that occur in the individual because of this vulnerability. These diseases, these problems are all around you, and probably at a frequency that you might find surprising. It turns out that about 22% of the population carry those HLA alleles. That means 40 million individuals within the US population. And this includes all biotoxins, meaning that what turns on this inflammation? Usually it's an organism or a fragment of the organism to some degree. So it can be everything from mold and elements from a water damaged building to infectious diseases such as tick-borne illnesses, viruses, parasites, even fungal elements, but also VOCs, MVOCs, bioaerosols. There's a variety of triggers that can turn on this immune response. And to clarify, biotoxin again means a living organism or a fragment of that living organism, like a cell wall, for example. But it does not include heavy metals and chemicals as far as we can tell. So those are classic toxins, and they certainly interfere with human biochemistry and can contribute to disease. But this is a separate category because of the ignition of the immune system and the vulnerability where these patients have poor antigen presentation. They remain stuck in that innate immune response. They can't turn it off because their genomics begin to shift over time, and it's a disaster for these patients. And in addition to that, one of the reasons why probably this has been so hard to diagnose is that not only are the proteomics, these inflammatory proteins, unique and therefore not often measured, but even when we have a suspicion of a biotoxin, for example, like Lyme disease, our tests are not very good. We know that in a variety of meta-analyses, actually, the two-tier testing for ELISA and Western blot tends to be relatively inaccurate and can potentially miss up to 70% of cases. And while the CDC has recognized that there are about 300,000 individuals with Lyme disease in the United States, that number is probably at least triple if we had better testing and surveillance. But there are diagnostic challenges, generally speaking, and that is to say, we first have to ask the question, does this patient meet criteria for CIRS? And we'll go through that process. More specifically, does this patient suffer from a tick-borne disease? So now we're having to rely on tests that, by definition, are unfortunately unreliable. And I'm certainly aware of all of the newer tests on the market, but many of them are not validated. And typically speaking, I think there's too high of a false positive rate. In addition to that, and think about this, let's just take a water damaged building, so mold and Lyme disease. Because of the proteomics, because of the transcriptomics, the symptom profile is essentially the same in these two patient populations. That in the data that we've collected and published, we've in fact tested patients to see, can they tell based on symptoms? Do they have a tick-borne illness or do they have a mold exposure? And it turns out they can't tell based on symptoms. And that is to say that the symptoms overlap so much because there's this uniform immune response to all biotoxins that patients can't tell what they've been exposed to. They know they're sick, but they can't use their symptom profile as a way of identifying the original exposure. And I know that there are whole surveys out there to say if you have these symptoms, it's Lyme. If you have those symptoms, it's Bartonella. If you have these symptoms, it's Babesia. Well, I wish I had a nickel for every time I had a patient who had been told they have Lyme disease, even though their Lyme test is negative, but they say, Doc, I've got all these symptoms. I must have Bartonella and Babesia. And it turns out that it's mold. And so that's where the genomics, the proteomics, being more particular and meticulous about your evaluation process, and I'm going to give you a path for that because it's really important to segment these patients and to know what you're dealing with. But sometimes it's very hard. Sometimes you don't know that, some of these tests are just not as reliable as we would like them to be. But there is a pathway forward. Even harder is if you come to the conclusion after all the testing that you've done that it's not a tick-borne illness, it's not a mold exposure, that it's one of the other 30 to 40 biotoxins that this patient is suffering from. So there are at least 30 entities that can turn on this inflammation, and many of them you might not have heard of before. Beta-glucans, mannins, spirocyclic dramines, lipopolysaccharides, actinomycetes, which we now know is a really big one in water damaged buildings, hyphal fragments, cell wall fragments, bioaerosols, endotoxins, MVOCs, VOCs, mycotoxins, you get the point. The list is long. It is notorious. We've even found that dental issues, periodontitis and bacteria hiding in the gums can also trigger this response. So you have to be a bit of a sleuth, and sometimes it's hard to discover what's turned on this inflammation in the first place. Even more difficult is the recognition that once the body's ignited, that if the patient's in the 20% and their gene response starts to change so that the inflammation becomes perpetual, if the patient is lucky enough to have gotten out of the moldy home or taken enough antibiotics or other antimicrobials to treat their infections, the genes don't turn off, that they can remain sick. And that can be confusing too, because it looks like there still must be a trigger around. And unfortunately, the body is taken over. It induces this chronic inflammation. They can't turn it off on their own. And who knows what the original inciting event was in the past. So we have to be really careful about, asking the questions, what turned it on? Is it still around? Does the patient meet criteria for this kind of specific inflammation? Do they fall in that 20%? And then what are we going to do about it? These are the questions that I always ask myself from a clinical perspective. And we do have a wealth of information now on these biomarkers. There are 25 papers in the literature. We've evaluated over 4,000 patients, including pediatrics. We've had multi-site confirmation. There's a variety of doctors that are certified in this whole approach. And we use a number of ancillary tests to help us confirm the diagnosis. There are changes that occur in the cardiopulmonary unit. This has to do with significant vasoconstriction that occurs in this patient population due to these changes in their proteomic profile. It can lead to restrictive lung disease. It can lead to interstitial lung disease, as well as, and this is a big one that I want you to really sort of absorb, the inflammation that's generated in this patient population not only is sort of noxious to the nervous system, but in fact, over time, these inflammatory proteins injure the brain. That we can see volumetric changes occur in the central nervous system as a fingerprint, due to the inflammation that has now resulted in essentially a brain injury. So you think about this: 20% of the population is potentially vulnerable. Once they establish this inflammation, it can create a variety of symptoms. This inflammation then triggers secondary and tertiary phenomenon in almost every body system: cardiopulmonary, GI, nervous, immune, the list goes on. But ultimately, this inflammation can actually damage organs and tissue. Maybe the most problematic is the brain itself, that this is a brain injury-based disease. And now we have some tools to measure that. It's called NeuroQuant. This is a volumetric brain scan. We can also apply transcriptomics, which we do. We have a genomics expert that we work with to evaluate gene response. We know there are very specific profiles that are expressed in sequences of genes that also act as a fingerprint, so that we know for sure now when a patient has been exposed to mold, when they've been exposed to Lyme, are they post-Lyme, did the treatments work? All of this can be read in gene reactions. This is not genetics. This is not, well, what genes do they have? It's genomics, and that is to say, with the genes they do have, what are the genes doing, which are on and which are off. In addition to that, there is a tremendous impact on digestive health. The enteric nervous system becomes very inflamed and dysfunctional. We see gastroparesis, gallbladder dyskinesia, motility disorders, chronic constipation, bloating, maldigestion, reflux. The GI tract takes a huge hit. In addition to that, and I'll talk about it in a little bit more detail, these patients develop gut permeability or leaky gut. And so they get this sort of secondary immune response, because now antigens are crossing the gut barrier, encountering the immune system, and triggering this sort of additional inflammation that makes people feel even worse. In addition to that, there are general immune changes that occur, sort of overexpression in certain areas of the immune system and under expression in others, so they're vulnerable to secondary infections. As I've mentioned several times now, the presence of these sequences on HLA with respect to the polymorphisms are where the poor antigen presentation begins. And then finally, we have some very simple tools to measure the inflammation, in particular brain inflammation, because that's key. This is really a brain on fire phenomenon. And one of those is called a visual contrast sensitivity test. And then finally, with all of this, we need to be able to target the changes that have occurred in those patients and correct those abnormalities. We are able to do that with our published protocols, which I'll go through in part 2, which ends with a very special therapy called VIP, or vasoactive intestinal peptide, which corrects the genomics, it corrects the volume changes in the brain. Unfortunately, this is a prescribed compounded neuropeptide. It is not something that's available over the counter, so the sort of crowning treatment is a prescription. We've not found a natural alternative for the ultimate step, but we have discovered, I think, some pretty reliable natural approaches pretty much all the way up until then. There are a few nuances along the way, though. Again, there are no differences, and I think this is really important, in the way that the body responds from a water damage building to really any biotoxin, including post-Lyme. And that is to say, whether it's mold or dinoflagellates or other organisms and pathogens, there is sort of this uniform but complicated response by the body. And therefore, the symptoms are pretty confusing in the sense that different patients can present with different symptom profiles, but the total symptom list is actually quite long. One thing that we've seen, though, is that symptoms tend to cluster. So we've been able to identify 13 clusters, and our diagnostic criteria includes if you have at least one symptom in 8 of those 13 clusters, then there's a high likelihood that this is a CIRS patient. We then go on to do our additional testing, occasionally pulmonary function tests with diffusion capacity, EKG, and stress echo. One of the main features of the cardiopulmonary phenomenon in this illness is vasoconstriction. So the small capillaries begin to tighten up, and that includes the connection between the heart and the lungs. So unfortunately, these patients are at risk for some mild to moderate forms of pulmonary hypertension. They therefore get some stretching of the right atria, and they're vulnerable to palpitations, vulnerable to the consequences of pulmonary hypertension, which obviously is a pretty serious illness. As a result of this peripheral capillary abnormality, the heart has trouble pumping blood to the periphery, and therefore they have declining VO2 max as well as exercise tolerance. These are patients that are short of breath. They get winded very easily with light activity, and they take a prolonged time to recover from exercise. And they'll talk about air hunger and general fatigue and shortness of breath. And I used to be a trained athlete, and now I can't walk from here to there without feeling like I'm totally deconditioned. So in addition to the inflammation, in addition to the brain-related changes, in addition to all of the digestive issues, they also develop these cardiopulmonary problems, which are pretty serious. We use the NeuroQuant, which is our volumetric tool to assess brain-related changes tied to a standard brain MRI. We're now able to run transcriptomics in this patient population to be able to really clearly identify when they're having an abnormal genomic response, and we've been able to sequence those genes and even now parse out what the original biotoxin exposure may have been. At least we can tell between mold and Lyme. And then we use all of this as a way to set the roadmap. So we need to correct these abnormalities as we go. The symptoms need to get better. Their breathing and cardiopulmonary status has to improve. We need to correct the brain-related changes in the abnormal gene response and get these patients back to normal. So here are 13 categories of symptoms. The hallmark is fatigue, but a lot of these patients have terrible headaches, migrating aches and pains, unusual peripheral sensations, light sensitivity, ocular issues, the shortness of breath and air hunger, coughing and sinus congestion, abdominal pain, secretory diarrhea, and or constipation. And I can tell you the effect on the cognition and mood is profound. So many of these individuals report short-term memory loss, word-finding difficulties, poor concentration and focus, even new fears and phobias, depression, anxiety, social withdrawal, sleeplessness, poor stress response, lack of coping skills and emotional resiliency. This is truly a brain on fire phenomenon. In addition to that, you get some more organic changes in the brain. So therefore, the hypothalamus is affected, which results in not only mood disturbances, but also appetite changes. So they develop a leptin resistance, and therefore they can't regulate their appetite. There's a lot of weight gain that goes along with this illness, in addition to temperature instability. So they almost look, they almost sound like to some degrees, sort of a really fully expressed and very sick version of a menopausal patient, or a male who's going through, who has hypogonadism because of the weight gain and the temperature instability and the mood-related changes. These patients can present in so many different ways, And, we go down these other rabbit holes and they get these labels of depression or chronic fatigue or fibromyalgia or adrenal fatigue or leaky gut or, all the things that we sort of hang our hat on to say, oh, I know what's going on with you. But then we do our therapies and they don't get better. And it's because there's this core issue of inflammation that's driving all of these other physiologic changes. And until you correct that, you can't get these patients better. So again, the cognitive and neurologic symptoms I think are really important to identify. And it's a systemic inflammatory response because of this innate immune activation. There's decline or changes in these regulatory neuropeptides, which I'll talk about in a little more detail. Alterations in other aspects of immune response, including Th17 cells, which express TGF-beta 1, which in turn creates imbalances in T regulatory cells. The labs that we've published that I'm going to have you go through, these proteomics, they're in peer-reviewed journals. We have 2 double-blind randomized controlled trials that support this. And now we've even, as I said, gotten into the transcriptomic side, which I think is really important in this patient population. So the labs that we use establish a baseline. They help us make the diagnosis, and therefore they're part of the protocol with respect to correcting each abnormality until we get to the end of the treatment process. And then VIP is the last step of therapy. But, and this is really important, even if the blood labs return to normal, that does not assure cure. And the reason for that is because the organs, and in particular the brain, still has changes that have to be dealt with. And there continues to be loss of the regulatory control at the genetic level. And that is to say we have to re-regulate gene response and fix the tissue damage. So this is really important. In addition to that, there are some other features of the illness that accrue as a result of the core condition. One of the most important is the potential presence of a staph infection that takes hold in the deep nasal passages, and it really only appears as a result of the state of illness of the individual. And as the patient gets healthier, they tend to be able to resist reinfecting themselves. But when they are vulnerable and inflamed and their immune system is not working very well, they can gather this special form of staph. We call it MARCoNS for short. That's an acronym. And we need to do the nasal culture and know whether or not the MARCoNS is present, and we have to treat it. We've shown in genomic studies that if MARCoNS is there, the final step of therapy, the VIP, will not work. So we have to make sure that we do that. So here's the evaluation process. I've tried to make it very clear in terms of, how does a patient meet criteria for CIRS? And the first is you give them the symptom questionnaire, you go over the 13 clusters, and you see whether or not they have at least 8 of the 13. Now, with that being said, sometimes the patients only have 5 or 6. That's okay, but they're starting to point in a certain direction. Then you do your laboratory evaluation. We have you order proteomic markers. These are very specialized neuropeptides and inflammatory proteins that you can get from actually standard labs. You can also do functional labs to help support the diagnosis, although they're not critical. Of course, we want you to draw the HLA sequences off chromosome 6. Again, standard labs will do that, nothing too fancy. And then you can also order a variety of infectious disease markers to see whether or not this is a biotoxin related to a pathogen. The patient then will also complete the visual contrast study. This is a 10-minute test that they do online. It's very inexpensive and quite reliable. It is a direct measure of optic nerve inflammation, which more broadly reflects brain inflammation. We have a 98.3% sensitivity of diagnosing CIRS if they have 8 of 13 symptoms in their symptom questionnaire for their clustering, and they fail their visual test. 98.3%. 8 of 13 categories for symptoms and failing the visual test. You don't even need to do the blood labs. That's how good the evaluation process has become over time. You want to do your H&P. You also, of course, want to do a detailed exposure history. That's your initial screen. Then you want to verify. So you look at all the results that come back. Tier 1, we need all 3. Do they have a known exposure? Sometimes that's hard. Have you done your differential diagnosis and ruled out other causes? Do they have positive symptoms in 8 of 13 clusters? Tier 2, 3 of 6. Did they fail their visual test? It turns out about 90% of people who are inflamed will fail their visual test, but 10% of people, no matter how sick they are from their exposure, they can pass it. So passing the visual test alone does not indicate that it's not a biotoxin illness. Do they have presence of the HLA? Are they in the 22%? And then we get into some of the blood-related changes. So here are a couple markers that you're familiar with and a couple that you might not be. The first is MMP9, matrix metalloproteinase. This is an inflammatory protein that's produced by macrophages. It breaks down connective tissue. We can also begin to see discordance in ACTH cortisol ratio, as well as ADH osmolality ratio. That's an interesting one because these patients tend to get hyperconcentrated with urinary frequency. It's a result of lower intrinsic levels of antidiuretic hormone, so the kidneys don't resorb water. Therefore, these patients are urinating throughout the day, and they concentrate their electrolytes, and so they get orthostatic issues. They get electric-like shocks when they walk across carpeting, and so this actually becomes a pretty big deal for them. So with your POTS patients that have fatigue and joint pain and so on and so forth, you really should look at CIRS as the model. And then finally, a low MSH, which is melanocyte stimulating hormone. MSH is produced in the anterior hypothalamus and is a master control hormone for a variety of processes in the body. So 3 of 6 of those, and then we confirm with at least 2 of 3 in tier 3, which is symptom improvement with treatment. They're ultimately able to pass the visual test, and then finally resolution of laboratory values. We then confirm the diagnosis and say, well, what is the original exposure? Is it Lyme disease? Well, you can do your 2-tier test. I prefer the NanoTrap. This is using new nanotechnology with a urine sample. We then have our patients swab their home. We want to know whether or not their home is moldy, so we have them complete the ERMI test, which stands for Environmental Relative Moldy Index. And then we can also, if they're really short of breath and we're worried about their cardiopulmonary status, they can do exercise tolerance testing. Almost all of our patients, if we can get it, they undergo a standard brain MRI. It's non-contrast, but then we add the NeuroQuant. That's actually a software program that many MRI centers have licensed. And then finally, the transcriptomics, which helps us really fully verify what the exposure is. So these are all of the sequences that I go through when I start evaluating the patients. I'm listening for symptoms, drawing blood labs, they complete their visual test, I see what comes back in terms of verifying the diagnosis, and then I confirm with some supportive tests to say, oh, it's Lyme disease, or no, it's mold exposure, or it's both, or neither. And then we look at their transcriptomics. So what is this visual contrast sensitivity test? Well, it was developed by the military over 40 years ago. It was designed to quickly assess whether or not a patient has had an exposure of some sort. It's very reproducible, reliable. There's quite good supporting data on it. It's just about the best marker beyond day 4 of a biotoxin-associated cytokine illness, meaning that cytokines are released like a storm in the brain. Once the brain becomes sort of chronically inflamed, we begin to see decline in visual contrast sensitivity. And what this really means is just color discrimination, that in fact people lose the capacity to distinguish between similar colors along a scale. In this particular test, typically patients are presented with gray circles that change their shade, and also there are very subtle lines inside the circle and they're pointed in different directions. So the patient just has to answer questions about these gray circles. It takes 10 minutes. It's $15, and there are some websites that are, I think, free as well, and very reliable in terms of whether or not is this a brain on fire phenomenon. And we've tested this in a biotoxin population. So here are some of our results in acute Lyme versus post-Lyme. And it makes sense that in both categories, 20% of patients fail the visual test, and it's because they have the presence of those alleles. This is what the result looks like. They cover one eye, they answer questions about the gray circles, then they cover the other eye and do the same thing. So they get a grid and therefore a total score. You can see on the bottom, this is a patient who's failed. And there's sort of a more detailed way of reading these grids to say, is it an acute exposure, chronic exposure, mold, Lyme, so on and so forth. But generally speaking, all you really need to know is whether or not they pass or fail. If they fail, and again, they qualify for 8 of 13 symptom clusters, 98.3% sensitivity that, yep, this is likely a biotoxin patient with CIRS. Here are those notorious alleles that I keep mentioning. These are the SNPs that occur on chromosome 6. 3 of them are what we call multisusceptible, meaning any biotoxin can trigger this inflammatory response with poor antigen presentation. We think some of them are specific to the type of exposure. There are several for mold, there's a couple for Lyme, there's one for dinoflagellates, so on and so forth. Now, we keep looking at this, and my suspicion is that the list is probably longer overall, and also some of the more specific SNPs are likely multisusceptible, like the 15.6 or the 13.6, for example. So, so with that being said, when you order this test, it's an HLA DRB DQB. It's typically reported in a way where it doesn't come in this kind of final presentation. You actually have to sort of reconstruct the alleles, and there's a Rosetta Stone to do that, which is fine. It's actually pretty straightforward. And you can find that on the survivingmold.com website on how to reconstruct the alleles based on the raw data that's presented on the lab report. So in addition to the visual test and the symptoms and the HLA, we also have our whole set of proteomic markers that reflect the sort of broad inflammation that's being generated within the innate immune system. The inflammagens that are produced might be ones that you haven't heard of before. C3A, C4A. Those are from the alternate complement cascade. So we're still in the innate immune system. These are proteins that are sort of triggered in sequence, C3, C4, C5, so on and so forth. But it turns out that the cleavage pattern generates very high levels of the alternate complements, not the standard complements. So C3, C4 will be normal, but C3 and C4A potentially can be elevated. C3A tends to reflect a bacterial exposure. C4A will often go up in just about any biotoxin illness. It's the only one, though, where we have so far found that there seems to be a difference in the level that's measured between patients with mold and patients with Lyme. That if the upper limit of normal for C4A is about 2,800, on average, Lyme patients tend to be in the 6,000 to 8,000 range, and mold patients tend to be 12,000 or above. Now, that's just a rule of thumb. I've had plenty of patients who have mold, and they're 5,000, 6,000. I've not had a lot of patients, though, that I can think of who have very high C4A levels of 15,000, and it's Lyme. It's almost always mold. That one is really pathognomonic. And then there's MMP9 that I spoke about briefly, as well as TGF-beta1, again, produced by Th17 cells. So there's a number of these proteomics that we're able to draw. And as they become more and more abnormal, we see these alterations in hormonal imbalance, total immune expression, vasoconstriction, immune suppression, so they get the secondary staph infection in the deep nasal passages, and sort of just kind of general physiologic chaos. So all of this is generated as a result of the exposure. In 80%, there's proper disposition and removal from the body of these biotoxins. But unfortunately, in the HLA susceptible, these biotoxins can directly injure nerves. So the degradation of the myelin sheath, of the nerve tissue in general, is pretty extraordinary. There's literally excoriation of the nerves. But in the genetically susceptible, when these cytokines are produced in the brain, this storm that occurs begins to affect various areas, including the hypothalamus. So there's destruction of leptin receptors in the posterior portion of the hypothalamus. This is why patients gain so much weight. And I'll tell you, if you have a patient that presents to your practice and they report fatigue, brain fog, uh, mood disturbances, sleep problems, and they describe significant weight gain, 20, 30, 40, 50 pounds in a few months, and they say, I haven't changed my diet, I haven't changed my exercise regimen, it's likely this condition until proven otherwise. That the weight gain associated with this illness is so extraordinary, so stubborn, so hard to get off, even when you detox them and you get them on low carb diets and do high intensity training, and they're like, I lost 3 pounds. And it's because the brain is on fire and the body just will not shed this weight. In addition, the anterior hypothalamus is also affected. And so we see alterations in expression of vasoactive intestinal peptide, MSH, which I've mentioned, and arginine vasopressin. MSH in particular starts to drop. So when MSH levels are low, there are so many problems that accrue as a result. MSH is supportive for melatonin production, so they have non-restorative sleep. MSH also is permissive for endorphin levels, so these patients have chronic aches and pains. Interestingly, MSH also controls tight junctions in the gut. So and when MSH levels are low, these patients can develop sort of gut permeability, or colloquially leaky gut. So that's why you can get this sort of secondary inflammatory response, because the gut is becoming like a sieve and toxins are crossing, the immune system is responding, and these patients are pretty miserable in that regard. In addition, MSH controls the balance between cortisol and ACTH levels, as well as ADH and osmolality. In addition, there's upregulation of the aromatase enzyme, so you get this overexpression of estrogens. And when MSH levels are low, this is permissive for that MARCoNS, this resistant coag negative staph infection, to grow in the deep nasal passages. These MARCoNS are pretty nasty bugs, and they contribute to this illness greatly. In addition to that, as capillaries constrict, we have induction of hypoxia inducible factor, which triggers vascular endothelial growth factor, as well as TGF-beta1. And so changes in the periphery, including capillary restriction, means that these patients have shortness of breath and air hunger, pulmonary hypertension, vasoconstriction, which throughout the system, which makes these patients lose exercise capacity and loss of VO2 max. And if that weren't enough, we see alterations in immune activity related to autoantibodies, in particular gliadin, cardiolipins, and diminishment of Treg cell pools, as well as production of C4a and C3a, which I've mentioned, and then finally MMP9, Pi1, so on and so forth. So it's just a mess. These patients are absolutely a mess. Their brains are on fire. They're having all sorts of symptoms. They've tried typically lots and lots of therapies, and nobody knows what's wrong with them. So we've had to sort of articulate an expression of this, and that is to say, you got to draw these markers. You got to measure an MSH. You have to measure a TGF-beta1 and a VEGF and a C3a and a C4a, MMP9. You swab their nose for MARCoNS. You measure the balance of ACTH cortisol and ADH osmolality and their HLA susceptibility, and you start building a picture, a profile of the individual. Symptoms, visual test, proteomic markers. I realize for some of you, maybe many of you, this is all a new language, but it's a new language I really want you to learn, because the penetrance of these HLAs means 22% of people that are likely walking into practice, maybe more, because we, of course, attract a very sick population who one of our primary complaints is fatigue. That if your treatments only feel like you're working around the edges, and you say, well, I've fixed their leaky gut, and I've supported their adrenal fatigue, and I've balanced their hormones, and I've given them stuff for energy and blah, 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 and they just, they feel held together. Maybe their symptoms are a little better, but they're just not all the way there, that I've not fully figured them out. Well, you got to look at this entity. I've also gone into detail for each of the markers. I'm not going to spend much time explaining them now. You can read about them, but I wanted to make it available to you so that you can look at them and read about these issues to say, well, you know, what is it that each of these markers do? What do they represent? The first few, the VIP, MSH, TGF-beta1, C4A, all incredibly important and central to this illness process, as well as some of the autoimmune markers, the imbalance in the stress response, VEGF, vascular endothelial growth factor, so important for blood vessel health. The anticardiolipin antibodies, this is a really important marker. And the reason why is cardiolipin is actually a lipid, which primarily lives in the inner cell membrane around the mitochondria. And as you have more epigenetic insult and abnormal gene expression, the ribosomes start to make not only abnormal proteins, but also there's a abnormal lipid expression in this patient population. So doing lipid replacement is so important for these patients. Phospholipids in general, and in addition, they have altered cardiolipin at the mitochondrial inner cell leaflet, and this can trigger an autoimmune response there too. In addition, ADH osmolality, MMP9, leptin. So what do these labs look like? Well, here's a normal CBC and even a normal CD57. You know, so sometimes CD57 goes down in this patient population, sometimes it doesn't. Does not seem to be specific to Lyme. I know people really want to hang their hat on that, but I can tell you, I got a lot of patients in my practice who have very low CD57 levels. Here's an elevated MMP9, and this one is instructive. So you'll notice that the normal ranges that we use, and I posted them for you, are not always the same as what's reported on the lab. So you can see for this one, less than 984, but this is an individual whose MMP9 is 501. Our upper limit of normal is 332. That's actually published in the literature, and the range should be changed on the lab, but they refuse to do that. So it's important to recognize that sometimes the reported range is the same and sometimes it's not. MMP9 is one of them. We do use the normal ranges for ADH and osmolality as well as anticardiolipin antibodies. Here's an individual whose TGF-beta1 was high, so 7100. One of the Lyme test C6 was normal, D-dimer was normal. And here's another one like MMP9 where the stated range is not what we use. You can see it says 0 to 40, but more importantly, the normal range is 35 to 81. And that's also for good reason too. In fact, 35 to 81 used to be the normal range. It's since been changed, but that's not the correct range. The correct range is above 35. So this is an individual whose MSH is low at 20. And here is a very high C4A. So this represents someone who's clearly got a mold exposure. It's not 6,000, which likely is Lyme or maybe mold. It's 17,000. And so that is specific to Lyme. So what are some of the microbes I want you to look out for? The mycoplasmas, tick-borne illnesses, some of the viruses like EBV, HHV6, and CMV, toxoplasmosis, which is really a parasite, HSV1 and 2, streptococcus, and mold forms. So Lyme is, you know, it's scary. We don't want to deal with this. How do we manage it? How do we diagnose it? Well, you know, there are lots of problems. You know, the symptoms are all over the place. There's lots of false positives, and conversely, the Western blot is poor sensitivity. But now we have an ability to do a little bit more careful data collection. And I prefer the NanoTrap test. This, I think, is a lot more accurate than the standard ELISA and Western blot. You can't really get false positives on this test. It's very highly sensitive. It's absolutely specific. It was developed at George Mason University in Virginia, and it was designed to be a better and more accurate test for Lyme disease. And it's easy because it's a urine test. And we're looking for an antigen in the urine. In this particular patient, this is an individual who was negative. How do we swab for mold in the home? Well, we do the Environmental Relative Moldy Index. This was originally designed by the EPA. We look at probably 30 or so different mold forms that might be pathogenic for the individual. Since then, there's a newer test called HERTSME that was developed. It's got 5 or 6 mold forms that we think is even more accurate. The HERTSME test and ERMI use PCR technology. And here's the result of the ERMI test. So anything above 2.0 is too high for a person who carries the HLA alleles. They're too sensitive. And this is an individual who swabbed their home and their ERMI score is 10.51, well above 2. And this is logarithmic. So every point above 2 is worse and worse and worse and worse and worse. 10 is really, really high. This is a problem environment. Here I've given a instruction sheet. What does this all mean for the individual? Because patients often have questions about it. You can also do some functional tests. Saliva cortisol is helpful, food sensitivity panel because of the leakiness, maybe some functional GI tests or microbiome tests. You can also look at lipopolysaccharides. Certain companies will do this for you now. Of course, that's a biotoxin trigger. DAO and mast cell activation. Lots of discussion about histamine release in this patient population. Even blood-brain barrier tests that we know that breaks down in these patients. But let's go back to the brain. So what are these executive symptoms telling us? Well, it turns out that there is damage to the brain as a result. Now, how do we measure those brain-related changes? NeuroQuant. This is a volumetric brain imaging study. We look at 11 key centers in the brain. They can change over time as a result of this inflammation. It was cleared by the FDA in 2007. And it's a standard MRI that's added to the non-contrast imaging study. Easy to do. Many MRI centers now license NeuroQuant. You just need to contact them, your local ones, and find out. There's even a website that you just Google, NeuroQuant centers, and a map will show up, and you can just put in your zip code and see if there's one nearby. You'll be surprised at how many are around. It was originally designed for Alzheimer's and Parkinson's to capture early brain-related changes. And we know there are volume alterations in the CIRS population too. Turns out that they can go in either direction in terms of volume increase or decrease, interstitial edema or atrophy and pruning. And that is to say, atrophy is true scarring. Pruning is dendritic retraction, where the brain starts to shrink to protect itself from this inflammation. But when you take the inflammation away, you can get restoration of that tissue. Dendritic pruning is a very hot topic. We see its presence in everything from PTSD and stress to multiple sclerosis. It is a result of this inflammation, but it also represents plasticity because once you remove the offending agent, those dendrites can regrow. This inflammation is primarily driven by macrophage activity with their cytokine production, and these are specialized macrophages in the brain called microglial cells. And there's a whole immune system in the brain that gets turned on because of this CIRS exposure. And again, it won't turn off on its own, and this is what is related to the brain-related changes. Atrophy is not pruning. Atrophy is actual real destruction of the tissue. It's like a scar. And just like when you cut your hand and the wound can scar over time, so too can the brain. So this is a normal slice. Then the software program begins to segment the slices. You get a general report. I like the fact that they give me the hippocampus because it will tell me, is the hippocampus too small? Usually that relates to stress. But I'm most interested in the percent changes in the left and right side or hemispheres of the brain. We look at key centers that reflect these alterations. We add them up and have a scoring system. And what was fascinating to us is that even though the symptoms are the same for biotoxin exposures and the proteomics are essentially the same except maybe C4A, the brain-related changes were unique. That in fact there was a fingerprint for mold and a fingerprint for Lyme, and they're not the same. So we see enlargement of the forebrain parenchyma, cortical gray matter, and hippocampus in mold, as well as atrophy or volume loss in the caudate. But the cortical gray matter, hippocampus, and caudate are normal in post-Lyme, and we see a small forebrain in putamen. What's fascinating about this is that the caudate and putamen are contiguous structures in the basal ganglia, and the caudate shrinks in mold, and the putamen shrinks in Lyme. This is our scoring system for mold. This is our scoring system for Lyme. And so once you see this illness, it's going to change your life as a practitioner. I guarantee it. It's going to open so many new doors and therapeutic options for you. You're going to say, finally, these patients that come prepackaged with the labels of chronic fatigue and fibromyalgia, and they're on all sorts of medications and all sorts of supplements, and they never get better. And you even dread seeing them. We all have those individuals. Well, at least a portion of them have CIRS, guaranteed. And when you do your evaluation process and you say, oh my goodness, I can't believe this person who I've been treating for such a long time, this is it. Because they failed their visual test, they have all the symptoms, they have abnormal MMP9 and MSH and C4A and TGF-beta1, they have the presence of the HLA alleles, and sure enough, 10 years ago when they were a grad student and living in some, a rented home on campus, and they were exposed to visible mold, and they said, oh, that's when I got sick, but I just thought it was from, the stress of studies or whatever. And you say, no, it was an exposure. You're in the 20%. Your body ignited this inflammation. It has a hard time turning it off. And we have to do some things to help your immune status get back to normal. And they go, this is such a revelation. I've never done these tests before. No one's ever talked about this. And you're going to get used to sort of having this conversation with people. And because so many of the symptoms are the very things we often treat in integrative medicine, the fatigue and headaches and mood disturbance and digestive complaints and hormonal imbalances and weight gain and all of this stuff. And you're going to find that maybe it's more than 22% of your patient population. And you're going to even question yourself saying, I can't believe so many of my patients have this issue. But, that's the nature of our practice. And so from my perspective, you still have to be so good at dealing with all those other common issues that we sort of talk about in our integrative and functional practices, the adrenal fatigues and leaky guts of the world and hormones and so on and so forth. And yeah, you have to detox your patients and get their diet right and get them moving again. But there's some core features to the therapy that are really going to turn these patients around, that are somewhat kind of unique to CIRS. And once you get those nuances, these patients will unfold beautifully for you. And they'll be so thankful. And you'll feel like a much more sort of complete provider in that regard. So there are a number of treatment challenges in this patient population. You have to be able to eliminate the exposure. You have to correct that inflammatory response, repair the injury to the nervous system, and restore normal genomic activity. All of those, all four, are foundations of how we get these patients better. It's not just one thing. It's not just, quote, giving lots of antibiotics for Lyme. That's not enough. It's not enough. You have to turn off their inflammation. You have to correct their secondary and tertiary abnormalities. You have to get rid of that marcons in their nose. You have to deal with the excoriation and injury to the lipid membranes and the myelin sheath, and turn off the abnormal gene response and repair the brain. Got to do all of it. But when you do, these patients get better. I also relate this to part of the general Da Vinci education series. Dr. Laval and I have talked about our triad modeling, and this entity of CIRS clearly falls into the category of gut immune brain, that these patients have sort of a fully expressed version of this triad abnormality. And this is where I see all of these labs fall with respect to this particular triad. And they're so sick and so compromised, and you have to do this kind of full evaluation with them. But as I said, they will unfold for you. You will not believe it. It's so straightforward. And in part 2, I will talk about how you deal with these patients therapeutically. It is a stepwise process. It takes time. It's months. It's not weeks. It's months and months to get these individuals better. But you can and you will. I promise you that. And I hope that this has been an eye-opening lecture for you. I hope this is new information. Take time to absorb it, review it, and start gaining some confidence to recommend a visual test, order some of these blood labs. And again, I think you're going to be pleasantly surprised at, finally beginning to solve some of these cases in patients where, nothing ever really seemed to work. And now you realize, hey, it's a CIRS patient, no big deal. Clean up the mold in their home, turn off the inflammation, repair the damage, and away they go, and they're forever grateful. So thank you so much for your attention, and I look forward to speaking to you once again as part of the Da Vinci Education series.